Okay, what I'd like to do, I had some prepared comments, and I think instead of using those is I want to go directly to questions and answers from the audience. First of all, summarizing our talks. Can you hear me in the back? I got a big mouth. Okay. It's clear to me that we're early in these epidemics in Australia versus what's happening in Israel and perhaps what's happening in California. Looking at Paul, what he showed in Australia, again, the beetle has been there for a very short time. He's seeing less damage there. What Stan told me about Israel is that this was a slowly developing situation in Israel. It's not been here in California as long as it's been in Israel. So even though we might have slightly different pathogens and beetles involved, my guess is that we may see, again, even in Australia and perhaps in Florida where I am, a developing situation where we'll have some more severe damage in the future. So in the remaining time that we have, I'd like to talk about several different areas. Diagnosis, Keith mentioned he's got a real-time PCR diagnostic technique. Management, I was going to talk about what we've been doing with laurel wilt. I've tried fungicide management. Jorge has tried insecticides, repellents, attractants. I've got a poster on the fungicide stuff. Imperfect solution for that. Cultural management, sanitation. For our disease, sanitation is very important. Is sanitation going to work for this disease? Resistance, disease resistance. We see very poor resistance in avocado to laurel wilt, and I'm hearing confusing information about whether or not. So experience in the floor on that would be, I'd be interested in that. Epidemiology of this. It looks like this thing moves very slowly. For us, one of the more pernicious aspects of laurel wilt and avocado is that this thing moves rather quickly via root grafts. It looks like this is going to happen with this fusarium situation, but again, your perspectives on that are welcome. The ecology of this thing, it sounds like, based on what Stan has found and what I've found so far, that this is a mycangial association, which always implies that there's a close, co-evolved situation there. Are all these fusaria moved like that? Are there phoretic associations? Any experience on that? Let's talk about that. Lateral transfer to other beetle species. Our first speaker, Dave, talked about this other species, Eulacea. We have really good information now that our pathogen, Raftelia loricola, is moving around laterally to a lot of other species of ambrosia beetles. So let's think outside the box. Is that possible with this situation? And what's going to happen in natural ecosystems? We're focusing on avocado here, which is really important. Avocado is a $350 million crop for a year here in California. It's really important. But what about natural ecosystems? Laurel wilt is really hammering native forests in the southeastern U.S. What is this problem going to do to coastal live oak and other situations here? So with that, somebody's got a mic. Please, Yuri's the first question in the back.
If you're going to moderate it too, we get better sound if you stand behind it.
Okay.
I'll just ask our Australian friends. I'm wondering. Thank you. I'm wondering how well is the native fauna of ambrosia beetles known in Australia? Because I'm wondering, you have a fairly widespread animal there that sometimes seems to pop out and cause damage, but maybe not often. And I'm wondering, it's native and common in Papua New Guinea. Is it possible that we don't have an invasive species, but actually an old species that just occasionally, now that we know what to look for, now we see some damage?
Possibly. Most of the research is in forestry, as explained before, so they don't really know. We haven't done much trapping apart from managed forests and things like that. And the landscape's so diverse that, it could be in there, especially the Cairns region. Maybe it's come down from New Guinea. It's actually existed in that region for quite a while. Southeast Queensland, probably not, because of the managed forests around there. They would have seen that in the trapping previously. So I'd say possibly half and half.
The Bovaria bassiana that you mentioned caught my ear. So Bovaria bassiana is a species complex, unfortunately. Is this true Bovaria bassiana or sensu lato?
100% matched
to a voucher Bassiana. So there, I mean, there could be, I suppose, and that was based on ITS or and EF. Okay, I'm just thinking about biological control.
Yeah, it seems to work like in that region, it works really well. So.
Yeah, I would follow it.
Follow up on that, we saw Bulgaria yesterday in one of those trees, and a bunch of dead beetles with a white fluff on them, typical symptoms of Bulgaria bassiana. So it's definitely, this is everywhere, and the bark and ambrosia beetles are very susceptible to it, so I'm not surprised.
Yeah, I would just follow it up. I have a colleague in Beltsville who works on entomopathogenic fungi, including Bavaria, and has done a lot of the work that Dave just alluded to. So I would highly recommend contacting Steve Rayner. And in the spirit of Randy saying, think out of the box, I'm on this one like way out of the box. But if you find that this isolate, whatever you want to call it, is a good biocontrol agent, I'm wondering, this is totally way out in left field, whether or not you could transform it to get it to express attractant so that the beetles would actually actively seek it, sort of like a Trojan horse. And if it's producing alcohol, I know you guys would like it.
The floor is open. Yuri again.
This is for Akif. You mentioned numbers of trees that species of trees with the fungus positively living in it. Do you have data on how many tree species support the beetle reproduction?
No, we don't have it. So that's the first thing that we are going to do to find out which species are being reproduction. Yeah.
I thought it was really interesting that in Australia, apparently the beetles go for small branches. So that's for tiny little ones, or?
So originally we only saw it in about 1.5 inch round, so
Yeah.
true woody branches, but recently those actually had to go looking specifically for them. Yeah, they're about centimetre, perfectly green branch, not really very woody at all.
So could you see it on the outside, or did you just have to cut it and then you saw the beetles in there?
Like looking for boreholes
Okay.
in the actual branch. So.
Okay, because that's, I think in here we generally find them in branches that are thicker. At least, I think Tim Thibault, you saw, what was the thinnest branch you found them in? 3 centimeters. 3 centimeters. So that's a little bit more, 1¼ inch or so. And so we never find them in real thin stuff, but just in, at least this thick. Okay.
Dave had a question.
This is a question for Akif. And one thing that I'm hearing a lot is, people are mentioning Fusarium solani, and I think that that can mean a lot of things, obviously. But looking at trying to guess from the sequence matches that you were showing for the things that were Fusarium solani, they look to me like they're in a part of the solani complex where you see a lot of endophytic fungi that are frequently, not always, but frequently associated with woody hosts. So I'm wondering I'm not sure what you did to surface sterilize, or if these were things that were potentially in the gut of these insects. Can you maybe comment on that?
So we did the surface sterilization on those beetles before we chopped them and crushed them in distilled water. So I have also been working on the other canker pathogens on avocado. I have never seen, or I actually put those other old Fusarium species in my phylogenetic tree. None of them match with these. So these are coming up with these beetles. Associated or not associated, I don't know, but that's how they come from. But our finding was like the 1 out of 10, I would say. It's like the very rare. It wasn't common.
I would expect to see them in the gut.
I'm curious, for those of you guys who are getting seriously impacted, what are the recommendations you're going to give producers about how to manage this problem? I talked to Stan, and he said, man, we're not Yeah, we're not giving any recommendations yet. These things eventually we're going to have to, at least for my situation, if I had a grower come up, you're going to have to say, geez, based on our best guess based on what we know, we're going to have to sanitation something other than, geez, I have no idea. Dead silence. Okay, thanks, guys. Yeah. Well, I mean, what about just the basic idea that sanitation could play an important role, and when you take that stuff out of the orchard, grind it up, you know? Jason's got really good information, at least for another ambrosia beetle, that chipping knocks the heck out of those things. We saw from our Sri Lankan colleague that they chip up tea branches. If you've got a chipper and the branches of the logs aren't too huge, to me, it's a no-brainer. Now, Ruben has mentioned that, geez, we grow avocados on steep hillsides here, and that's going to be difficult. But, you know, when you have that opportunity to chip stuff up, boy, I think it'd be a good idea. Ruben, yeah.
Well, I think initially we're going to have to find some symptoms that we can show growers to recognize what it is they have, rather than just talk about we have this problem. And for example, the dieback is, if we can just indicate to them, look at this, the way this thing, and then confirm, because right now Akif gets phone calls all the time. Well, we saw some sugar leaking out here, and it turns out to be the wrong thing. It'll never happen unless we have some kind of methodology of communicating. So it seems to me like if there was a way to show photographs that some growers can access, some kind of database that to show where, how to recognize. What I found, we invited people to come for this meeting tomorrow afternoon. Very few growers really registered to come, and I recommend to them to bring their field people that,
Yeah.
you know, I don't think they're really ready to see what's going on.
You need to educate those people who are in the field daily seeing those problems. And I'll just do an aside here. Jonathan Crane, our extension tropical fruit guy in Homestead, he's done a lot of work on this laurel wilt stuff. And what he's done is he's developed these nice color brochures that he hands out to those guys and say, boy, this is what you need to look for to distinguish laurel wilt from everything else you might see that kind of mimics it. Yeah, well, are those going out to producers and their field people? I mean, Akif is, yeah, go ahead, Akif.
So Ruben is right. So there are other pathogens also cause the similar symptoms like the branch dieback, such as root rot or branch canker pathogens like the Botryosphaeria group of fungi that we have been working on that. So, and also Phytophthora citricola, which has the new name Mangii, also causes similar, like the sugar volcano on the trunks. So, the first thing that we start with those handouts to distribute to our growers, and it's the good idea that to have the field days with the growers to show them what the symptom look like in the field, like we did with you yesterday. So now, I mean, you can tell from your experience, it is amazing to see by yourself in the field than looking at the picture in the fields. Yes.
Picture is a first step, and you know, the caveat there is this is what it looks like. But then, if you got serious doubts, take it to Akif. Akif has got this diagnostic now that can say, "This is the fusarium I got out here." Now we're much more sure about that situation.
Randy,
Jason, you want to?
Yeah,
please,
I was just going to say maybe Akif can learn from our mistakes with laurel wilt when it comes to molecular diagnostics. I mean, we thought we had a good PCR assay based on a ribosomal DNA sequence. It was very highly specific and sensitive. We searched GenBank to make sure there was nothing in there that it would amplify. We tested a bunch of other Raffolias, and then we got a sample from an avocado tree that lit up positive. And the cultures morphologically couldn't really be distinguished from Luricola, but we were suspicious of it, and Randy did pathogenicity tests and found that it wasn't, in fact, Loricola. And when we did further phylogenetic analysis, we determined that it was an undescribed Rafflesia that was extremely closely related to Loricola but non-pathogenic. So we had to abandon typical loci for diagnostic purposes and go to microsatellites because of that. And it caused a lot of problems because the avocado growers were, upset because unfortunately the information was released. That's a whole nother story, but it got out that this was a positive, and they took steps because they thought they had it in a commercial grove, but it really wasn't. And so I guess, you're lucky in that you have these fusarium experts that have a lot of, isolates to work with and make sure your assay is really, really tight. But, we're working with these pathogens we know very little about, and so it's that kind of thing can happen.
Stan Freeman up front here, please.
Randy, just to reply to your sanitation advice. In Israel, the beetle goes straight to the trunk. There's no way that we can advise any sanitation whatsoever.
Well, but let me just hold that thought then. If this thing moves slowly downwards, you'd want to avoid the possibility of that moving to the next tree via root grafts. So I'm not, by sanitation, I just don't mean take out branches. If you know for a fact it's gone down in the trunk, take out the whole tree, do some stump grinding.
Problem is that when you have an infected orchard, it's usually affected at every level with every tree being infected.
Okay.
So that's the problem. You have to deal at the trunk level. And that means injections and just about very difficult to deal with fungal pathogens that are harboring or colonizing at that level.
Hold the mic. Yuri's got a comment, but I just wanted to ask, since I think it was Zvi that brought up what do you guys know about cultivar resistance in avocado? Is there tolerance to this?
Well, we can talk about resistant varieties and less resistant varieties. We cannot talk about, I would say, not resistant, susceptible and less susceptible. We don't talk about resistant or immune varieties of avocado at all.
Okay, yeah. Yuri.
If you guys are done with this topic, I just wanted to say that I have developed a small, simple online method for growers and everybody who's not an expert to tell if their red bay or avocado tree has been killed by the red bay ambrosia beetle. And it's really simple. You go on my website and you answer 4 simple questions. How much the tree has wilted? Let's say, many or all branches. Is there this sticky brown thing coming out of your trunk? Yeah, something's coming out. Are there the sawdust noodles? Let's say, sure, yeah, I can see them. And how about if I peel the bark? What do I see underneath? There are some streaks. And so you answer those questions, go down here, you push submit, and it'll give you the answer. In this case, yes, it's most likely the red bay ambrosia beetle. In other cases, it would be no. So if anybody's interested in adopting this little tool, it's not public yet, this is a prototype. So I'll talk to you guys about how to actually upload it and make it functional. If anybody's interested in adopting this for the California situation, let me know. We can work together.
What happens when you put in no noodles show up? Because what we're seeing in avocado is you can have trees decline and you have a hard time, even a red bay, have a hard time finding that evidence for ambrosia beetle involvement.
So this works on the principle of summing up all the answers. So if all the other symptoms point out to the fact that it's probably a red bay ambrosia beetle, even if you don't see any noodle, it'll still tell you that it's probably the beetle.
Our colleague from Sri Lanka, I apologize, I forget your name. Yeah. Right behind you, right there, yeah.
I have a question for Akif. It appears to me the survey records only the presence and absence of infestation in a given location, whether the infestation is there or not.
Most of the time, infestation be confirmed by the fungus, new fungus developing.
Okay, my question is there any possibility that you can quantify the infestation in a given location? How severe that infestation in a given location? On top of your survey on yes or no, whether it is possible to say if it is yes, how severe is it, that, right?
Yes, I didn't show those data, but when we find the infested plants, we took all the data. So where the infestation from trunk or main trunk or secondary branches, or what is the infestation level, we count them. Yes, we got all data. Yes.
Yeah, I was wondering if you had some dollar figure for the losses caused by this disease in Israel. So I think that would help the California growers to bring awareness here in California. So I don't know if you did some economic studies about this, or if you are doing some studies, but I think dollars talk a lot more sometimes. So.
I just want to, reflecting on Jason's point about molecular diagnostics, I'm a little bit concerned about overselling that. We're clearly still in discovery phase with respect to new Fusarium ambrosium-like pathogens, and any molecular diagnostic method is going to be dependent on our current knowledge of the diversity that exists. And every time we look, we're finding new things still. So, and I don't want to discourage people from developing methods like that, but you have to be very careful about over-interpreting the results. And at this point, I think a false positive is worse than a false negative. And the potential is very much there that you could find something new, get a positive, and it's not a real path, or it's not really part of this problem.
Point well taken. I was just going to follow that up. Is, you know, our experience is mainly with diagnostics for Fusarium head blight, and I have a colleague, Todd Ward, who's developed SNP-based assays, and what he's done to compensate for our lack of knowledge about the diversity of the pathogens, which is always going to be a factor, is develop clade-specific probes so that you can say that it's a member of this group, and then have taxon-specific probes, but build in redundancy so that you can try to compensate for your lack of knowledge about the pathogen diversity. So that's worked quite well. So where we found, for example, a pathogen that's gives a positive genotype for clade, but doesn't match any of the species probes, then we can be reasonably sure that we've encountered something new.
Ruben had a question.
I'd like to ask Jonathan Crane, maybe he can give us a little bit of input of what he does, and maybe we can follow some. And there's right next to him sits a PCA who is going to be I'm sorry? Okay, I'll shut my mouth.
It's his dime, Mary Lou. It's his dime, so.
So there's no, just so I get an answer, there's no economic studies being done in Israel, or there is? Not yet, okay.
Mary Lou has given us 5 more minutes if there are additional questions or comments, or everybody's exhausted. Okay, I'm not going to beat a dead horse. Thank you very much for your attention, and thanks to the speakers.